I think it's more there. And of course, the thing about science is that you need to take evidence and form your own critical analysis using evidence. And that's why referencing in quality publications is so important. So, another technique is case control studies, and this makes perfect sense. You know, you find a bunch of people with whatever it is you're interested in, and a bunch of match controls, and you compare the two. Case control studies. And what you do is you detect your SNPs, and then you literally you can do a simple table, not so simple now because you've got hundreds of thousands of SNPs, but if you're looking at just the one, you just do a simple table and do chi-square. Not anymore because it's pretty complex, but you just literally look at the frequency or the distribution of the SNP that you're interested in one group versus the other. And if 60% of the alcoholics have this particular SNP in the alcohol dehydrogenase gene, but 30% in the control, well then that's a big difference. So that's a predisposition SNP. You're more susceptible to alcoholism if you have that SNP. Doesn't mean you're going to get it. You may never drink. In which case, you're never going to be an alcoholic. So possession of SNPs doesn't mean that you're going to get it, and that's why we have to be careful. Because if you happen to go into health journalism, or medical journalism, and you translate papers badly into newspapers, you can worry the hell out of the public. It's so important to read papers carefully and interpret them properly. Okay, so just some stats there. So that's how you do it. You find the people, you define exactly what it is you're going to study. And you might think, oh, that's easy. But it's not. Because, okay, if it's type 1 diabetes, they'll have pretty strict, symptoms and diagnosis. You're either a type 1 diabetic or you're not. But what if you're studying depression? That's not so easy to define. It's variable. Different people define it, differently. Different patients will fill in questionnaire differently. Different clinicians will have different opinions about who's depressed or not. Not easy. And if you get it wrong, then these studies, if you get the design of the study wrong, or if it's inaccurate, or if it's not that good, it doesn't really make much sense. So you define the people carefully, collect them, find them, inform consent, there's all these documents to sign, get their blood, usually, or saliva these days, but it can be blood. Extract the DNA, store it in the freezers, and you've got enough to work on for years to come. Cases, controls. You've got to collect a huge number of them to make the study valuable. And then you start looking for the SNPs. And then you try to define which are the disease-specific SNPs, literally by their distribution. If this group happens to have more of a particular pattern of SNPs than that group, by quite a big way, that's saying something in terms of the predisposition. You have to be careful. You find one particular SNP, and it's 90% in your type 1 diabetics and 10% in your control, and you go, oh, I found it. That's me, done, my career, I'm sorted. Plain sailing from now on. That professorship, I can see it now. But that may not be the disease-causing gene. Remember, they're in linkage disequilibrium literally. It may be the one close by. You can't assume that you've found the disease. There's more work to be done. It could influence another gene. It could be a marker for a region of the genome. So we have to interpret it could be linkage disequilibrium. And as I say, linkage disequilibrium will come up more as you go on in your course. But it's based in things where SNPs are found together more than you'd expect by chance. You expect from mum and dad, because you know about meiosis, you expect a kind of a heterogeneity in terms of the mixture of the genetics from both of them. But if you find a certain region that is there more than you'd expect by chance, then they're in linkage disequilibrium. And that's because they're usually quite close together. And you know a recombination in meiosis, if they're so close together, then they can be displaced by recombination. So,